Hello, everyone. Just waiting. There's a lot of people joining right now, so I'm just waiting for everyone to be fully connected there. Okay, I think we'll slowly get started here. So hello again. It gives me a real pleasure to introduce our speakers for the last CIRC seminar of the year. So today we'll hear from Nicholas Raun and Dr. Jamie Kramer. Nicholas obtained his bachelor's from the University of Alberta in 2016 and his master's in 2019 from, from Western University. His master's was done under the supervision of Dr. Kramer, and Nicholas had won an award for the best research thesis within his department. In 2019, Nicholas moved with the Kramer lab to start his Ph.D. at Dalhousie University, where he's now completing his thesis. I hear that Nicholas is a very talented student. He works with complex imaging methods, bioinformatics, and his thesis focuses on the role of Trithorax group histone methyltransferases in brain function. So I'll now turn it over and let Nicholas tell us about his work.
Wonderful, thank you. I'll share my screen now. Awesome. Okay, yeah, thank you all to CIRC and for giving me the opportunity to present on my research topic about trithorax and its role in neurons and long-term memory. So in our lab, we study the trithorax group of epigenetic regulators, which are highly represented in neurodevelopmental disorders and includes complexes such as Phi-SNF and COMPASS. My project focused on the COMPASS family of histone methyltransferases. So I will be talking to you more about that. So with the goal of understanding COMPASS complexes in neurons, our lab opted to use the fly as a model system. In the fly, there is a brain region referred to as the mushroom body, which is responsible for learning and memory. This brain region is also largely dispensable for life. So we were able to make some pretty dramatic genetic changes there and then see the consequences of that in neurons in live brain tissue. Additionally, because it is the memory region, it has a relevant biological output when you consider the role of these complexes in neurodevelopmental disorders. With that in mind, we assayed courtship memory in the mushroom body using 2 RNAi lines to target the different enzymes of COMPASS. So when we look at the SET1 and TRR branches of COMPASS, we see that there is a short-term and long-term memory phenotype. However, knockdowns for trithorax and its cofactor menin-1 did not affect short-term memory, but did have an effect on long-term memory. Because of this unique phenotype, we decided to pursue studying this gene a little further. And then given the role of trithorax in tissue specification and development, we first asked whether this long-term memory phenotype was due to developmental effects. And instead, what we saw was that trithorax is dispensable when it comes to long-term memory during development, but is required in the adult. And that is true also for menin. Additionally, we saw that the morphology following a ubiquitous or a continuous knockdown in the mushroom body also has no effect on the morphology. Now, a key defining characteristic of long-term memory, at least in contrast to short-term memory, is that it requires memory-dependent transcriptional activation and new protein synthesis. Given that trithorax is an epigenetic regulator, we first asked whether or not it could be involved in memory-dependent gene activation. So we took trained control flies and compared them to their naive counterparts to identify memory-induced genes, and then found that, in fact, these genes were also induced following trithorax knockdown. As you'll probably notice, what we did also observe was that there was a significant increase in the baseline levels when we consider naive and trained trithorax knockdown flies. Going back to these basic molecular functions in long-term memory, we wondered if trithorax could possibly be regulating them as a group and contributing to the phenotypes we've been observing. So when we considered all genes annotated for translation and transcription, what we found actually is that as a group, translational genes are downregulated following TRX knockdown when we consider initiation and elongation. However, transcription is unaffected. We then suppose that the transcriptional overactivation we saw for memory-induced genes may be a compensatory mechanism for a loss of translation in the mushroom body. Therefore, we asked whether or not it was possible that translation was actually disturbed in trithorax knockdown flies. To assess this, we used the FunCat system, which allows us to label newly synthesized genes or newly synthesized proteins and compare them between different conditions in a tissue-specific manner. In such a way, we saw that trithorax knockdown flies have reduced labeled proteins, which indicates to us that translation is in fact reduced. With this result, we were then confident that we could start building a model. We found that trithorax regulates translation, and translation is an important component of long-term memory. However, we do know that trithorax is an epigenetic regulator, so it must regulate the transcription of some genes. So we sought out to identify these target genes that may be impacting translation and long-term memory. To do this, we went back to profile the methylation patterns in the mushroom body following trithorax knockdown, and we identified a number of trimethylation and monomethylation peaks. We then compared and contrasted these peaks with differential expression. Again, lysine 4-methylation is an activating mark. So we looked for genes that had reduced expression following knockdown, and we were also fortunately able to find trithorax chip profiles that were previously published from cell culture and found that there is an intersection between all three of these methods at least 6 genes, which I'm showing you here. And we consider these to be direct targets of trithorax in the mushroom body. What's interesting about these genes is that when we compare their expression in the mushroom body to the whole head, we find that they are especially enriched in the mushroom body. This suggests to us that they are involved in some critical mushroom body component or functions and may actually be required to define the mushroom body cell identity. I will note, although I don't show the data here, the increases in expression you're observing are similar to genes that are generally considered to be identity genes like ILIS. We also were able to validate at the protein level for 2 of these genes that they are in fact highly expressed in the mushroom body. Here we see HR51, a transcription factor, and LDH, lactate dehydrogenase, which is a critical metabolic enzyme. Going back to the behavioral output of the mushroom body, we see that 4 of the 6 target genes also impact long-term memory, like trithorax. So with all this data in mind, we can go back to our model and expand it out further. We now know that trithorax regulates at least 4 genes, HR51, MFS3, LDH, and DGP1, which have various biological processes which may be impacting long-term memory. DGP1 is a known translation enzyme and has been implicated in learning in rodents, so that's an easy conclusion to make. However, we first wanted to ask how HR51 could be affecting long-term memory. Thankfully, again, there were published data sets from a chip experiment in cells for HR51, and we found that the binding profiles of HR51 are adjacent to or overlap with the binding profiles of trithorax and are adjacent to the transcription start site and methylation that we observed in the mushroom body. Additionally, HR51 bound to more translation genes, which we identified as downregulated following a trithorax knockdown. With this in mind, we can further build out our model. HR51 regulates a subset of trithorax target genes, and additionally regulates some downstream translation genes, which we believe to be impacting long-term memory. With that portion of our pathway filled out, we next wanted to develop whether or not metabolism was involved in long-term memory of the mushroom body. Thankfully, in recent years, there has been a lot of work and publications revolving around the role of lactate and other metabolites in memory. Specifically, lactate is required to provide the energy for translation in following memory induction. And additionally, that was in rodents. And additionally, in Drosophila, it's also been shown recently that there's an upregulation of metabolism following memory induction. So with that in mind, our job was then to link trithorax to the regulation of these metabolites. And to do that, we used the FRET systems Laconic and Pyronic, which allow us to measure metabolite levels in a tissue-specific manner. Through this, we were able to show that lactate and pyruvate levels were not different following a knockdown of trithorax or LDH, which was expected because LDH was primarily expressed or primarily found at the protein level to be in the gamma lobes. And in fact, when we looked in the gamma lobes, we saw that trithorax and LDH knockdowns resulted in a decrease in lactate levels and an increase of pyruvate. Therefore, trithorax and LDH are responsible for maintaining a pool of lactate in the gamma lobes. The question then for us was whether or not this pool was required for translation. And indeed, what we found was that when we knock down LDH, we see a reduction in translation similar to what we had found originally with trithorax. Taking these results, we went back and built out our model further. We now observe that trithorax through LDH is able to regulate the pool of lactate in gamma lobes, which we propose to be providing ATP necessary for long-term memory required translation. There is a few other pathways here which we want to fill out, and that's currently work ongoing, but we are rapidly reaching the conclusions that trithorax targets an epigenetic network of novel mushroom body cell identity genes involved in metabolism and translation. In such a way, trithorax then fulfills a classically understood role of neuronal identity specification in a novel manner. So that's as opposed to neurotransmitter identity or action potential rhythmicity. Additionally, this is also the first direct evidence of how COMPASS genes promote expression in neurons and how those genes impart cell identity. Specifically, we've shown that trithorax impacts metabolic capacity of mushroom body neurons, which is specifically required to mediate long-term memory. With that, I would like to thank my supervisor, Jamie Kramer, other members of my lab which contributed to this work, as well as our funding agencies and these labs and stock centers which have provided the reagents required for my work. Thank you.
All right. Thank you, Nicholas. That was very nice work there. So we'll open the floor for some questions. I'll ask that you please use the Q&A function instead of the chat function. So you can go ahead and type it in. In the meantime, I guess I'll start with one, Nicholas. So is there data also showing an involvement of K4 demethylases in long-term memory, or perhaps also in the regulation of these metabolites like lactate levels?
So for your first question, I don't know specifically. I haven't seen any publications on that. Can you repeat your second question for me?
Yeah, so I was simply wondering if there was any evidence. So if COMPASS is regulating or helping regulate some metabolites to then regulate memory, is the same true for the demethylases? Do they have some impact in the process?
Yeah, so actually there has been work in our lab revolving the demethylases. I believe they're still unpublished, but there are some of them that are necessary for long-term memory. I don't know whether or not they're also required for short-term memory, though. I'll have to look into that again.
I think we have a question in here. So from Siamak, can you elaborate on the novel neurons that you mentioned, Trithorax, regulating, affecting?
Right. So so what I was referring to was they're regulating a novel set of genes in neurons that haven't previously been identified or haven't previously been worked on. So these are specifically the memory regulating genes in flies. And we've shown that they regulate a set of metabolism and translation genes that which has not been shown really for any of the COMPASS enzymes. Yeah, I hope that helps answer your question.
All right, we have another question from Jacek. Very nice. I was curious about the specificity of Trithorax. How many active promoters, what proportion are targets of Trithorax?
Right. So we haven't actually done chip on RNA polymerase. We tried, and it didn't turn out that great. We weren't able to create good libraries, so we weren't able to sequence that. But that's a very good question. We do have ATAC-seq data, which I'm currently working on contrasting. Basically, the best shot we have is that we can look at open promoters or promoters with open chromatin, and hopefully that indicates that, although it won't say definitively that they're active promoters, at least they're accessible. So it's likely that they could be transcribed in mushroom bodies.
Okay, the next question, Shujie. Very nice story. Can you show your first slide again? My research focuses on SETDB1 oncogenic function in cancer immune evasion. Can you talk about how SETDB1 functions in your system?
So where I believe this is the slide you're talking about. If not, there's also the data slide. So I've only done a screen so far of these with on Courtship. We've also done, we have some published data on this, and we've shown that other behaviors are largely unaffected, specifically in a mushroom body knockdown. So there's some specific effect in memory that we want to explore. And actually, I'm starting a project now just to do sort of comparative bioinformatics on all the COMPASS components. So we've done a lot of mechanistic studies with Trithorax, but now we want to find target genes in the mushroom bodies for TRR and SET1 as well. So I'll be work forthcoming and hope I can answer that question in better detail later.
All right, and we have one last question here from Hassan. Did you check histone lacylation marks if they're up, down, given what you said about lactate?
Yeah, so that's a good question. That's one we've actually considered and talked about in the lab. From my understanding, and there might be new literature that I haven't seen yet, but it's not been done in Drosophila. So it could be a lot of work before you can get to the point where you can competently create sequenceable libraries in Drosophila. It is something I think is worth doing, specifically given our work, but not something that's been done yet, and not something I'm currently planning on doing, but would be worth seeing.
All right. Thank you again, Nicholas. Some very nice work there. So next we will have Dr. Jamie Kramer. Jamie obtained his Ph.D. in 2005 from Memorial University, where he ran a functional analysis of Drosophila FOXO transcription factors. He did his postdoctoral training at Radboud University Medical Center in the Netherlands, where he developed Drosophila models of brain disorders for conditions caused by mutations in chromatin regulators. Jamie became assistant professor at the University of Western University in 2014, while further holding Canada Research Chair in neuroepigenetics. He's now associate professor in the Department of Biochemistry and Molecular Biology at Dalhousie University, and his lab focuses on understanding the molecular mechanisms of memory and the role of chromatin regulators in memory and brain disorders. So, without further ado, please welcome Dr. Jamie Kramer.
Thank you. Thanks, Eric. Thanks a lot for the introduction. And I'm just going to share my slide here. Okay. Does that look okay? Everybody can see and hear me. Anyway,
Perfect.
Thanks so much for the invitation to speak here. I've been attending a few of the seminars, and they're really exciting. So really happy to be a part of the seminar series. So I'm going to talk today about making long-term memories. So when I think about long-term memory, I think about, things going back to when I was a kid, like maybe that big trip down to Disneyland where, the whole family piled in the car and drove down to Florida or whatever to visit Mickey Mouse in the Magical Kingdom. Or, other things such as perhaps when you came face to face with a really cute goat that happened to be called Fred. These sort of things stick with you for a long time. Or a long-term memory can also be something more negative, like something really sad or traumatic. But in any case, these are really personal things, memories. So it makes it really exciting to understand and research how they work at the molecular level. So there is a lot of research on this, on long-term memory specifically. And a lot of what we know is outlined in this simple signaling pathway diagram. Essentially, in the formation of memory, a lot of several different possible signaling pathways can be activated. Notably, CAMP and calcium signaling are prominent pathways that converge on the transcription factor CREB, CMP response element binding protein. Many people have probably heard of CREB. So in response to neuron activity and memory formation, the idea is that CREB gets activated by the convergence of these signaling pathways. It binds to CREB response elements and activates genes that are required for memory. And the interesting fact about this is that as Nicholas mentioned, this gene activation by CREB is absolutely required for long-term memory, but it's not required to form sort of short-term transient memories. So we know all this. There's extensive research done in multiple different organisms, but it's still a bit of a question about how transcription actually helps form memories and what genes are activated. How does that work? This is still a black box. So in the presentation today, I'm going to show some work that we've done trying to understand better how this actually works, long-term memory formation downstream of transcription. So the fly memory neurons that we study, as Nicholas mentioned, are present in the mushroom body. So this is showing the mushroom bodies in the context of a whole fly head. You can see sort of a fly's face here on the screen. And the outline, the black line here represents approximately what the fly brain might look like within the fly head. And then you have the mushroom bodies, which are located here. And they're complex structures with multiple lobes. Nick mentioned a bit about the gamma lobe, which is particularly important in courtship, in the type of memory we study, which is called courtship memory. So just take a minute to explain what this is. So courtship memory or conditioned courtship suppression is based on the courtship behavior of male flies towards females. And here you're watching a male fly courting a female. You can see him buzzing his wing out. That's known as the courtship song. So he's singing to her and he's following her and he's trying to be able to mate. So this male in particular is a naive male that's never had another social experience before. So he is vigorously courting this female. What he doesn't know at this point is that that female has already mated recently. And female flies that have recently mated, they don't want to mate again. So this female is going to reject the courtship attempts. And over time, the male learns to not court mated females. The assay works by pairing a naive male for 7 hours with an unreceptive female. And then 24 hours later, we test the trained male's courtship activity. And we can see here that he's not really paying that much attention to the female. So the male has learned that there is no benefit to courting a pre-mated female, and now he's reduced his courtship appropriately. Okay, so this is the memory assay that we use. It was developed in 1979, so it's been around for a while, and it's a well-established form of fly memory. We do these assays in these large chambers with many different fly pairs, and we're using an automated video analysis setup to track the courtship behavior of many fly pairs at once. So the assay is semi-high throughput. And one great thing about this assay, that in the context of this known molecular mechanisms of memory, we know a lot about what's going on with courtship memory. For example, we know that the rejection aspect of the memory is relayed through dopaminergic neurons that interact with dopamine receptors and activate CAMP signaling. And it turns out that the male flies can recognize, they learn to recognize the smell of a mated female pheromone called CDA. And that smell is relayed through olfactory neurons that end up causing activation of neurons and influx of calcium and activation of calcium signaling, also converging on Krebs. So these pathways are conserved in courtship memory as they are in other types of memory in flies and other organisms. We also know exactly where this all happens. So the two signals that I spoke about, the dopaminergic rejection signal, these neurons actually synapse at the gamma lobe tip of the mushroom body. So that's all known. And then the olfactory neurons, they end up, well, not the olfactory neurons themselves, but a secondary neuron set that's activated in response to those synapse at the dendrites of the mushroom body, gamma neurons near the cell bodies. So these two signaling pathways, they converge on Krebs in the mushroom body. And so we've done a lot of transcriptional profiling, specifically in the fly mushroom body, over a time course of long-term memory to get at what genes are activated and important for that process. So to get the mushroom body neurons, we use a method called INTACT that we adapted from Henry et al. It's called isolation of nuclei in a tagged cell type. So here's a fly brain with the mushroom body nuclei labeled. We can lyse the brains, and then we get a mixture of cells that are either mushroom body nuclei or not nuclei, not mushroom body nuclei, and we can immunoprecipitate the mushroom body nuclei out with an anti-GFP antibody, and we get our purified mushroom body nuclei sample. We've been successfully done RNA-seq on these samples, as well as attack sequencing, and we've also done, as Nick showed, histone modification chip-seq on purified mushroom body nuclei. So this is our method that we're using. I'm using this throughout the talk to isolate mushroom body neurons. We also did a lot of analysis on the whole head sample. So when you first lyse the nuclear extract, you got a whole head sample, and some of the data I'm showing in the talk compares whole head to mushroom body to get an idea of what's specific to the mushroom body and what's more of a broad response. So student in the lab, a PhD student, Spencer Jones, did this long-term memory time course RNA-seq experiment where we collected samples at several time points during the memory assay. So like I mentioned, the training period is 7 hours where the male's paired with a female. So we collected samples at 3 time points during the training and several time points post-training, because post-training is when it's suspected that some of the memory consolidation occurs. We also collected naive flies at various time points during the day to account for time of day variation. And the first thing that we did with this data set is we looked for actually candidate courtship memory IEGs, or immediate early genes. So in the memory signaling pathway, well, it is relatively not understood how the transcription works or how it's important for memory. We do know a lot of the immediate early genes that are activated right away. So for example, CFOS is a famous one, but there's a lot of other ones that are known, for example, from this genome-wide identification of neuron activity-related genes in Drosophila. And so we took all the known candidates, about 17 of them, and we looked to see if they were induced during courtship memory. And we found two, HR38 and SR. So HR38 is an ortholog of the NR4A family of nuclear receptors. And you can see the data here shows gene expression, RNA-seq data from the trained mushroom body or the naive, and then the trained whole head sample or the naive whole head samples. And you can see here clearly HR38 is massively induced at the onset of training. And it starts to go down towards the end of training. It's then slightly reactivated post-training after the separation, and eventually goes down to baseline. And we see the same trend for SR, where it's activated early and remains activated post-training and eventually goes down to baseline. So these, we believe that HR38 and Stripe are now novel courtship memory IEGs. We also knocked them down in the mushroom body and demonstrated that they're required for long-term memory. So if you look at this, this learning memory data, you can see in controls, the naive flies, they court almost all the time, and after training, they court less. Whereas in the HR38 knockdown flies, there's no change. Sorry about that. Then in SR, we see a similar thing where the controls learn and the SR mushroom body specific SR knockdown flies, they don't. So HR38 and Stripe are induced in the mushroom body in response to memory training. They're required for memory. They're also genes that are massively accessible in the mushroom body. So we looked at our mushroom body ataxia data. Sorry, I'm losing my voice. And we found that these are ataxic profiles, and we can see that, for example, for SR here, the accessibility around the TSS is very high. The comparator here is genes that are highly expressed in the mushroom body, so they're already very accessible, and the accessibility is off the charts for SR. Also here, that happens to be an alternative transcriptional start site. And HR38, again, the accessibility is off the charts compared to other highly accessible genes. Also, at these accessibility sites, so here we're looking at our mushroom body specific attack data compared to published Krebs ChIP-seq data. And you can see that Krebs peaks really overlap with the mushroom body specific attack data. So Krebs looks to bind to HR38 at TSS and also at an internal site likely in a cancer. Again, at SR, we also see Krebs binding at ataxic peaks at the promoters, within some introns at likely enhancers, and also at the alternative promoter here, or at the alternative transcriptional start site. So you take all this together, these genes are highly accessible, they're bound by Krebs, they're induced by memory, and they're required for long-term memory. So these are now previously unidentified memory mediator genes for courtship memory. So we have starting to fill out the model of how this works beyond Krebs. So Krebs likely targets HR38 or SR and other memory-induced genes. So we also wanted to understand more about generally what's going on with these memory-induced genes. So that's what I'm going to talk about next. I'll just remind you, we collected these naive samples at different times of day, and so that we could compare a naive fly to a trained fly and identify genes that were differentially expressed in the mushroom body or in the whole head sample. So this revealed a lot of differentially expressed genes, and there's a big overlap between the two groups. There's also large amounts of genes that are specifically induced in the mushroom body or the whole head. And the expression trends of these three different groups, whether they're in the overlap or the mushroom body specific or whole head specific group, is interesting. And I'll show that now. So for the genes that are induced in both samples, so the overlap, we see in both the whole head and the mushroom body, there's a big induction at the onset of training that reduces and remains active at post-training for some time before reducing down to baseline. But the response here is very similar. So we expect that this is sort of represents a general neuron activation that occurs when a male fly is paired with a female for the first time. The different responses, so for genes that are induced in the whole head only, we see an interesting response where the divergent expression patterns happen later in the training phase. Now, what's also interesting about this group of genes in the whole head is that they appear to cycle in a circadian rhythm. So if you look at the dotted lines, which are the naives, they go up, down, in a nice 6-hour pattern, which is reminiscent of some circadian oscillations. And so it almost seems like these circadian genes are disrupted due to the interactions between the male and the female. And this would make sense in the whole head because the eye is a big circadian organ, and that's a big part of the head, which we're analyzing here. Now, in the mushroom body, it's quite fascinating because here we see genes that are induced high at the onset of training. They come back down almost to baseline, and then they're reactivated specifically in the mushroom body. So in this case, in the whole head, we don't see the reactivation at this time point, this later time point towards the end of training and after training, after the male has been separated from the female. And the timing of this is very interesting because it overlaps with what we expect is the time point of memory consolidation. Now, if you think about short-term memory and long-term memory, short-term memories are formed here in this time course within an hour or so, but long-term memories aren't. They only form after 7 hours training. Actually, we know that 5 hours of training can also be enough. So the consolidation of the long-term memory likely starts happening sometime in the latter half of training and likely occurs past the training period also, but we don't really know how long it takes or when it stops. But in any case, this mushroom body specific transcriptional signature here really overlaps with the consolidation of memory period. And the functions of the genes there are also quite relevant to memory consolidation. So just looking at a gene ontology heat map, if you focus on the mushroom body only sample, so these are the genes that are induced by memory only in the mushroom body. Down here, you see the enrichment of signaling pathways, all of the memory, known memory signaling pathways. So calcium, dopaminergic synaptic transmission, G protein signaling pathway, CAMP, protein kinase A. These are the real known memory genes, and they're induced during memory consolidation, specifically in the mushroom body and not so much in the whole head. So it seems like a memory-specific process. Additionally, the metabolism is quite interesting. A lot of metabolic genes are regulated by memory. But when you look at the types of metabolic genes, the ones that are enriched specifically in the mushroom body at this late phase of memory consolidation are related to TCA cycle and ATP production, which, as we know, as Nick showed in his, talked about briefly in his talk, we know that the mushroom body has a specialized energy requirement that's needed during memory consolidation. So this type of metabolic activation of genes really fits with what we know about the biology of memory and about the biology of the mushroom body in general. These memory-induced genes are also highly bound by the memory transcription factors that we identified. So SR, HR38, CREB also all have a lot of binding sites in these genes. That's more than threefold enriched over random chance. For SR, for example, more than a third of the memory-induced genes are bound by SR. And there's also a lot of overlap between the memory-induced genes that these 3 transcription factors bind, indicating that there's probably some redundancy in the response. But when we look, we found a really interesting trend when we looked at genes that were bound by more than one of these transcription factors, as opposed to the specific ones that were bound just by Krebs or SR. So when we look at memory-induced genes that are bound by 2 or more transcription factors, that includes pretty much all of the genes found by HR38, because HR38 is mostly in the overlap. But in any case, these genes have a very similar pattern when you compare the whole head expression to the mushroom body expression. If we look at one transcription factor, so bound by Krebs only, we see that the transcriptional responses diverge a bit. They have a similar pattern, but the level is much higher in the whole head. The most interesting result on this slide is the genes that are bound only by SR. So these are the genes that really have this mushroom body specific response. So you can see they're induced, again, high in the mushroom body at the onset of training, and they have this pattern where they are reduced and reactivated during consolidation. In the whole head, these SR bound only genes, they just reduce to baseline. So it seems like SR is somehow potentially governing the mushroom body specific late consolidation transcriptional signature. An interesting thing is that in the mushroom body, so this is mushroom body specific attack-seek profiles. In the mushroom body, genes that are only bound by SR, they happen to be less accessible than genes that are bound only by Krebs or bound by more than one of these IEGs. And so this raises the question of why are these genes less accessible? And perhaps there's something to do with chromatin remodeling involved in getting these genes activated at the late consolidation phase. So this model is developing a little bit in the sense that we now think that Krebs activates these IEGs downstream. They contribute to a broad early response that's elicited by courtship of the male to the female. The genes involved are metabolic and synaptic. Then there's this later mushroom body specific response that is characterized by memory signaling pathways and ATP production. And it seems to be governed by SR. Of course, as I mentioned, these SR bound genes, they seem to have low accessibility compared to the other genes. So we asked the question if chromatin remodeling factors are involved in this. And this is no coincidence because one of the big focuses of the lab is understanding the role of SWI/SNP chromatin remodeling complexes in the brain. This shows the Drosophila SWI/SNP complexes. A lot of people in the audience are probably familiar with these. They're ATP-dependent chromatin remodeling complexes. In humans, many of the members or components of these complexes are implicated in intellectual disability and autism. And actually, this protein complex is the most overrepresented protein complex in ID and autism. So it's really one of our goals in the lab is to understand what this complex does in neurons. And so we wanted to see, like I said, if it had a role in this memory-induced transcription that we've been characterizing. So we did a series of experiments where we knocked out BAP60. BAP60 is a core component of all SWI/SNP complexes. It's SMARCD1 in humans. And when we knocked down BAP60 in the adult fly just one day before the training period, so there's no developmental role, we actually, and I can't explain this, but short-term memory is increased. And so don't ask me about that because I can't explain it. But importantly, long-term memory is gone in these flies. So short-term memory is not reduced, it's increased, and long-term memory is gone. So it suggests that SWI/SNP might indeed, and BAP60 in particular, might indeed be involved in long-term memory gene expression. And so Spencer, again, in my lab, he did an experiment where he compared naive and trained flies in control strain and the strain that has mushroom body specific knockdown of BAP60. We did the mushroom body specific intact, we did RNA and attack sequencing in all of these samples. And for this experiment, the collection was done 1 hour after the training period. So at this post training time point where we do see a lot of gene induction still. And that's right in the middle of the memory consolidation phase. And the results are quite clear. So when we look at genes induced by training, there's 128 in the control and only 17 in the BAP60 knockdown. 2 of these 17 happen to be SR and HR38, so it doesn't affect those. If we look at sites of accessibility, there's 593 sites that increase accessibility, or sorry, 593 genes that show increased accessibility at 1 ataxic peak or more. And there's only 2 of these regions in the BAP60 knockdown flex. So clearly BAP60 is required for any sort of training-induced gene expression or accessibility changes that occur post-training. So in total, there's 79 genes that have a defect in training-induced accessibility and expression in the BAP60 knockdown mushroom bodies. And we can see what the expression looks like. They're clearly induced in controls from naive to long-term memory trained. They are induced at BAP60 knockdown samples. They're not induced by training, and they're also slightly reduced in expression overall. And at the chromatin level, we can see here the attack-seq profiles of the BAP60 knockdown, naive and trained are low, and then the control naive is slightly higher, and the control trained is much higher. So SWI/SNP is required for post-training changes in gene accessibility and expression. It's quite interesting because of these 79 genes, and I proposed that SR was involved in this late response, 35, so almost half of the binding sites of these genes, sorry, have SR binding sites. And of these 35, almost half of those are in fact transcription factors, and some of them are transcription factors that are already known to be important in the mushroom body. So it's almost like the mushroom body is saying, look, we're going to do memory now, so we need to reinforce that by activating genes that are normally activated in the mushroom body and reinforcing its identity as a memory tissue. And clearly, SWI/SNP and BAP60 have an important role in that. In fact, when you look at broadly at just big groups of genes that are annotated for things like learning, memory, cognition, etc., and we look just generally, so the control, naive to trained, you see you see an induction of these broad categories of genes, and then in the BAP60 knockdown samples, this induction is completely gone. So BAP60 is definitely, and SWI/SNP is definitely having a larger role in any sort of memory-induced gene plasticity that occurs after the training period. So what it doesn't affect, I mentioned this briefly, is HR38 and SR. So these 2 genes are completely normally induced in controls and in the BAP60 knockdown. So we believe that SWI/SNP acts downstream of these initial IEGs. Here, so you have Krebs activating the IEGs. BAP60 comes in at some point to open chromatin to allow for this mushroom body specific late memory consolidation response, which likely involves the activation or reinforced expression of additional transcription factors that are known to contribute to mushroom body memory capacity. And so just to highlight the main advances here. So we're starting to get at the fundamental mechanisms of long-term memory because we are diving into the black box that is downstream of Krebs in memory formation. So this really hasn't been elucidated in any detail as of yet. And so that's what we're trying to get to. Secondly, we think the mushroom body memory transcriptome that we've made and characterized is really a valuable resource to understand the role of chromatin regulators in brain function. And like a big focus of my lab is on chromatin regulators that are known to be involved in neurodevelopmental disorders like intellectual disability and autism. And there's a large number of genes that are chromatin regulators that are involved in these diseases. So we think this is a really nice system to interrogate their function in the context of cognition and gene expression. And finally, our results show that SWI/SNF, at least BAP60, is really required in acute memory processes. Not that they don't have a role in development, they do, but in the disorders that they're involved in, they are heterozygous mutations, and people live long lives, and so they're not necessarily developed or disorders that result from major defects in development. We think there's an adult component that could potentially be treated. So this finding that SWI/SNF is important in adult acute memory processes is relevant potentially for therapy for the disorders down the road. And so with that, I want to thank Spencer. Pretty much all the work I showed was done by a single graduate student, Spencer Jones, who is a great grad student. He will be on the market for postdocs this year, so keep him in mind. Also, I want to thank Nicholas. I'm really excited about his work and the project that he showed. He's also going to be on the market for postdocs, so please keep these guys in mind. They're both really, really good students. And I'd like to thank all the rest of the members of the lab and the funding, of course, and all the places that we got reagents from. And yeah, with that, I would like to take questions.
All right. Thank you, Jamie. We now know a little bit more about fly courtship. More importantly, memory formation. All right. Okay. So we do have questions coming in. So we have one from Jim. Great talk. HR38 and SR genes. Did you look at histone H3 serine 28 phosphorylation and K27 acetylation along these genes? Is LTM compromised when JIL1 is silenced?
Yeah, so no, we didn't look at these marks at all yet. So like we haven't, not at all. And I'm not sure what JIL1 is actually, but we did not, so I wouldn't know about that either.
All right. Annie is asking, does the loss of BAF60 disrupt NBAF or CREB or SR binding to memory genes?
That's a good question. So the loss of BAP60, so we don't actually, so what does it do to the SWI/SNF complex itself? I mean, that gets at your first question, does it disrupt NBAF? In flies, I don't know if there is specifically an NBAF, but I think that loss of BAP60, we know that SWI/SNF complexes can form still if that component is gone. So I believe that the complex is, and I don't have direct evidence for this, but based on what's shown in mostly cancer studies, is that loss of BAP60 doesn't cause the complex to fall apart or something. It just does not get targeted to specific genes properly. So it's a hypothesis, but we believe that BAP60 is required to interact with some transcription factor, potentially CREB or SR, to open up these genes. But it could also be the reverse, where BAP60 somehow and SWI/SNF opens up chromatin, allowing SR to bind. So we don't know the answer to these specific details.
All right. Jim is specifying that it's the JIL1 is equivalent of MSK1 too.
Yeah. I'm not sure. I don't know if those have been assayed in memory, to be honest.
All right. Shu J is asking, would you please talk about how calcium participates in the memory regulation and what is the readout for the calcium signaling? So the calcium signaling, calcium influx can activate, it's mostly through CAMK2, which can then phosphorylate a bunch of things, including CREB. And so, yeah, we haven't been looking at calcium signaling per se, like the people do calcium imaging, and it's there is a lot of that done in the context of courtship conditioning.
to show like what neurons are activated and things. But yeah, we're not looking at calcium signaling ourselves per se. So that was mostly based on previous knowledge.
All right, and that is commenting on this being a great resource. Question: what the final crucial genes and processes are determining memory is open. We are excited about sleep. Could sleep be disturbed in BAF60, etc., and then inhibit consolidation?
That's a good question, Annette. Yeah, I think you're right. Like I don't know, the final crucial genes and processes determining memory might be open, but I mean, we did find activation of genes involved in critical memory processes. So I think it seems like the gene activation is just what we see is it's bolstering known functions of memory downstream of my pathways. Yeah, no. Okay, then no. Could it be that sleep disturbances are involved? Yeah, I guess it could be. So we've done sleep. We didn't do sleep assays on the BAP60s yet, but we should. We started recently to do it on everything we tested in memory. We would also test sleep and activity. And we found like for a few chromatin regulators, just very mild changes to sleep, but those could still be important. So it's possible that it could go through something to do with sleep as well.
All right, Annie is asking, do you get neuronal mistargeting in the memory circuit neurons in BAF60 knockouts?
Yeah, so in some ways we do. So like the data I showed today is adult knockdown just one day before the training happens. So we don't see we don't see developmental defects then, although we've also been studying SWI/SNF in mushroom body development. So the mushroom body goes through a whole extensive axon extension and remodeling process through the earlier stages of fly development in the larvae and then in the pupae. And we do find, I wouldn't call it mistargeting, but defects in remodeling. So normally the gamma neurons, actually at a certain point in development, the axons get essentially chewed away and they regrow for the adult, and that process is defective in the BAP60 knockdowns and other SWI/SNF knockdowns as well. So yeah, SWI/SNF is important in this neuron remodeling that happens during pupal morphogenesis as well in the mushroom body. But right here, we're in this study, we're skipping all that by excluding the knockdown to adults.
All right, and we're smack on time. All right, so we'll leave it at there. So I'd like to thank again our speakers. That was super interesting. And this very nicely concludes our CERC online seminars for this year. Stay tuned for an announcement in the January edition of the CERC newsletter regarding the next seminars for 2023, and they will be held at the same time slot, but every second month. So in the meantime, just wanted to wish everyone a happy holiday season.